The potential to personalize medicines based on our unique gene composition is rather novel to all of us. But today is the opportunity for us to understand this better. So today's agenda is as follows. We will start with our esteemed keynote speaker, which I'm completely honored to welcome, Steve Tomlin. So he will be presenting to us about innovation and personalized medicines. Steve Tomlin is the Director of Children's Medicine Center, Associate Chief Pharmacist at Great Ormond Street Hospital. And once Steve has completed presenting his session, we are then welcomed by Dharmisha Chauhan. She is the lead genomic pharmacist at North Thames Genomic Medicine Service Alliance. And she will be talking to us about why we cannot ignore race when it comes to pharmacogenomics. As always, you will see a questions tab in your chat. Please use this to pose any questions that you'd like to ask for our speakers. If time is unpermitting, we will compile these questions for our speakers and upload them onto our digital meds optimization futures page. I will add the link to that in the chat as well. So without further ado, I am absolutely honored to welcome Steve Tomlin to talk to us about innovation and personalized medicines. Steve, if you may.
Thanks very much. Can you hear me?
Yes, I can hear you perfectly fine. Thank you.
Thank you. It's an honor to be with you today, and I'm actually on my phone. It's amazing that we can sort so many amazing things out with medicines, but when it comes to IT, there's still a few things which don't quite always work, but I hope everybody can hear me. And just to reassure you, whilst I am pediatrics through and through, this isn't primarily a discussion about children. It is about personalized medicines, but I want to start with pediatrics because I think it shows a real link as to, I guess, where pediatrics has always been thinking and where it fits into sort of today's practice. So I've set up recently, in fact, the last year, a children's medicine center, and it is primarily to think about personalized medicines. And medicines for children have been a problem for a long, long time. And in some ways, we've been talking about personalized medicines since we started with children, because we can't give them all the same dose. And I'll touch on some of that. Next slide, please. So why have we always had to think about personalized medicine for children? It's because there is no such thing as a standard child. And it might seem a bit strange, but on the whole, we do think of adults as being primarily the same. And that is sort of, they're somewhere between 50 and 150 kilos generally, 3 times variance in weight. And since most dosing that we look at is based on weight, which is probably wrong in the first place, but that is a factor, we get away with it. And I use that term fairly loosely, but I think it's really important that for so many years, we have got away with it with medicines, i.e., and it's why, you know, if you look at tablets, most adults are on 1 or 2 tablets of most drugs. And that's because drugs on the whole have got a fairly wide therapeutic range. It doesn't mean they all get the same result. It doesn't mean they all get the same response in terms of positive, and it doesn't mean they all get the same side effects. But we generally get away with it. And the concept, particularly for prescribing and for drug production, makes it easy. Because if we had to think of every patient as different, how would we provide the medication that are needed? And I guess that's where pediatrics comes in. We've always had to think about that. You know, there's a 200 times variance in weight, sort of 0.5 kilos up to 100 kilos. And therefore, just on a weight basis, it's impossible to give them all the same dose. That's not fully personalized medicine, but it is a start. And it's, I guess it's been in our mindset for a long, long time. And because of the complexity of that, on the whole, medicines haven't been made for children, giving us products that aren't appropriate. You know, you can get 10 or 100 times one dose out of a vial. And you know, if in a neonatal unit, the project that's on that slide there, neonatal unit, you go to the cupboard and take out a vial, and for 5% of all doses, you can get 100 times the dose out of one vial. Well, that's asking for trouble, and it's often done. And the biggest worries in pediatrics are the fact that we give 10, 100, even 1,000 times overdose, and it's almost there to happen, the old Swiss cheese effect. And so we have to think, how do we need to improve this? And it also means that most things don't get licensed for children because it's really hard to do studies in so many cohorts. And it goes to, you know, back to neonates. 80% of drugs used in neonates are unlicensed. It's only 2% in adults. Therefore, the evidence base is very, very different. But are adults going to have to start getting used to the same as we've been getting, sort of dealing with the pediatrics for such a long time? Next slide, please. So personalized medicines, it's about what makes somebody different and responding differently. And again, these are some slides for pediatrics, but we're starting to understand that, in fact, we've got to think about this for adults as well. So, yeah, I guess the one which I always love is the graph at the bottom with diazepam, a drug that's been around for a long, long time. That's the drug that sort of has helped people sleep overnight, Valium, and it's got a half-life of sort of 10 to 12 hours in older children and adults. In a premature baby, it's got a half-life of about 80 hours, which means if you give a dose, it's still hanging around a week later. Therefore, really important that we understand variances in patients. So that's obviously a liver enzyme variance, but there's clearance via the kidneys. There's also gastric absorption. But there are so many other things which make individuals respond differently. Next slide, please. So just going back to what it means when we have all these variances, I mentioned that most things aren't licensed for children. In fact, every drug that gets a license in Europe by the EMA, only 14% of them are licensed for children. And at the end of that patent, so after 12 years, it's still only about 25%. Does it matter? Well, it doesn't matter in as much as, you know, we can use drugs all the time without a license. What a license does give, though, is some reasonable assurance about the fact that some work has been done on its efficacy and its safety profile. But I think we all know it doesn't mean it will be efficacious in an individual patient, and it doesn't mean it will be safe in an individual patient. It just gives you some assurance about work that has been done. And I'll come on to why that assurance isn't really what we need as we go through. So next slide, please. So just a few sort of stats to do with pediatrics and what it means not to have licensed medicines, and the fact what we're dealing with is a certain amount of, or has been a certain amount of guesswork. And I guess the only one that I'm going to read out is the bottom one on here. Children are 3 times more likely to have an adverse drug event than an adult. That's quite a statement. But if we haven't got the same sort of evidence base when we're using medicines, in some ways, not surprising at all. You know, we get adverse drug events with adults. And a lot of that is probably because we haven't got the dose quite right. It doesn't mean the drug is wrong, but have we got the dose right? And so these things are happening all the time in pediatrics. And so, do we need to start thinking about this for adults? Because I think it's going to become a real key theme for the future as we get personalised medicines. Next slide, please. So I think there's been two things which have really got me thinking over the last the last few years, and they are both really associated with genomics. And I think genomics has brought a different thought process to medicines, but in fact, it's only highlighted something which really we've been dealing with for years, and it's the fact that not everybody responds the same to medicines. And genomics is just another part of the mindset that we've got to get ourselves around. We need to understand pharmacokinetics. We need to understand pharmacodynamics. We need to understand pharmacogenomics. And all of them play a part in an outcome of a patient. And therefore, whilst at the moment, because it's all quite new, genomics is seen very much as something different that we've got to get out to do and look at. In 20 or 30 years' time, we're hopefully talking about it exactly the same as we talk about pharmacokinetics, because that's exactly, in terms of drug use, exactly how we need to be thinking about it. We need to take all of these factors into consideration when we're looking at dosing. The two, I guess, that we've really been thinking about for a while, certainly ototoxicity from gentamicin. For years and years and years on the neonatal units, we've been worried about hearing loss with gentamicin. It's one of the first drugs that most premature babies get. And there's something about, well, if we know that ototoxicity is actually a genetic issue, how do we test and when should we test? We've been able to do genomic tests for aminoglycosides for the last 10 years, but it's always taken sort of 1 to 2 weeks to get a result back. That doesn't help on a neonatal unit when it's one of the first drugs you give after birth. And I think what is changing now is technology and that ability to be able to test and to test quickly. Just in the last couple of years, a machine has been developed which can actually test for that genetic variance within half an hour. And as soon as you get to that stage, you get to a point of thinking, well, that now can change whether I will or won't use a particular drug in a certain circumstance. The other was codeine, a drug that was been around for a long, long time, and we've been using in pediatrics for a long, long time. And then all of a sudden, there was a MHRA alert saying basically we shouldn't be using codeine in children. And it was primarily a genetic issue, and it was a hyper metabolizing it, and people getting very high peaks of morphine. But it's actually a minority of people with that genetic change. And therefore, if we understand genomics, genetics of a patient, there's no reason why we can't use it in a majority. Now, whether we need it back or not, and in fact, it's low-dose morphine just as good as codeine. There's all sorts of clinical discussions around a drug like codeine. But in fact, it's about understanding, and if we've got data in front of us, we can prescribe accordingly and appropriately. So in my mind, they're the two things which I think really started getting me thinking that we need to have a different approach to medicines practice. And almost two of the reasons which have driven me to set up the Children's Medicine Centre at Great Ormond Street really started thinking, how should we be looking at good practice of medicine use? How should we be looking at working with the regulators to get licenses? And how do we really think about both effective and safety of medicine use? Next slide, please. So we do need to start thinking about personalised medicine and thinking of everybody as an individual. And it's let's say genomics has sort of almost started this talk and some of the themes, but it's there for kinetics, it's there for dynamics, and it's very much putting everything together to ensure that we look at people as individuals, that they won't all respond to a medicine in exactly the same way and have the same outcomes. But how do we really do that? I think this is the key to sort of getting this right. How do we move forward with a system that actually requires either different medicines or different doses for individuals when we've got an industry that is based on standardization? And hopefully we will never have to lose that completely. But we need to understand when it's appropriate to use. Standardization is great because it's one of the biggest safety features that we can bring into medicine's practice. And just talking about a drug where you've got loads and loads of different doses possible, it's what we've been talking about in pediatrics. If an adult turns up at the pharmacy counter with a prescription and say, I don't know, if it's for say something like ranitidine, and it says 150 milligrams twice a day. Well, that's the dose that was licensed. It's the dose in the BNF. And you go over, crossing over to your shelves and, oh gosh, the tablets come as 150 milligrams. Very hard to get it wrong. Virtually all tablets are made so you take one or two. And you generally don't ask the weight of the patient the other side. Pediatrics has always been round the other way. Because it's not standard doses, it's 2 milligrams per kilogram is the standard dose for ranitidine. And so you've got a parent there, and you've got to check the weight, and they say, well, the weight is, they're 6 months old and they're 6.5 kilos. 2 milligrams per kilogram, I need 13 milligrams. Well, the liquid used to come as 15 milligrams in a mil. So what does the prescriber put, and what do you dispense? Because you can't actually draw up 0.8777 recurring mils. And so it becomes really interesting about what is the right dose. Is it still covered by the license? 2 milligrams per kilogram is. Therefore, somebody is supposed to draw up and give 0.877 recurring mils. So really interesting to see how all of these bits fit together and who should or shouldn't round something. Obviously, the easiest thing to do is round it to 15 milligrams, which is 1 mil. But is that actually covered by the license anymore? Probably not. But does anybody care? Clinically, probably absolutely fine. But would it be the same for another drug? And it's understanding the kinetics, the dynamics, and everything else that goes with it. So we need to think about medicines in a very different way moving forward. Next slide, please. So where have we got all of our data from in the past? And is it actually the right way of looking at things? It's always been that evidence-based practice is really looking at things like RCTs. The pinnacle of best research is doing a randomized controlled trial. But a randomized controlled trial gives a patient a definitive dose. And you end up with a curve that whether someone was treated and what side effects they did have. But within a curve, there's always, not everyone's right in the center of that curve. Therefore, some might have got some benefit, but not as much as maybe somebody else. But you don't get that data from a randomized controlled trial. A randomized controlled trial gives you one dose. So let's say we're going to give everybody 5 milligrams. Well, some of them might have done better with 4.7 milligrams and all these sorts of things. Therefore, a randomized controlled trial only tells you what it was set out to tell you. But is it the right way of thinking about medicines? And instead of evidence-based practice, is it that we should now be thinking about practice-based evidence? Far, far more important. Of course, you've got to start somewhere. And how do you determine where you're going to start? But if you've got a drug that's being used you almost totally need to do then is collect real data on real patients and work out what is happening. And this is where data lakes and real world data come into their own. And it is the only way forward. NICE have accepted that it's exactly where we all need to be going. And real world data is being used all over the world now. And there are millions and millions and millions of data sets that can be used. In Eden, which is a European data set, I think already, and it's just touching the top of the iceberg, there's over 800 million data sets of drug use within that data lake. And this is where we need to start getting our data from. And therefore, you almost want everybody to remain in a trial. If you're on a drug, we need to collect that data. Therefore, every aspect of anything that happens to a patient on a drug should be carried on. In cancer, it's what's been done for a long, long time. So certainly within pediatrics, if you've got ALL, you join a trial. And every few years, something tweaks in it, see if it can be improved. But it's the same trial, ongoing. And you almost cannot be treated for ALL within pediatrics unless you're in that trial and you remain in it. But we should be doing that for all medicines. It's that ongoing data which actually tells you far more than the original RCTs did. Next slide, please. So real-world data is the way to carry on looking, but somehow we need to work out what the original doses should be. And if you're going to try and do that in loads of different populations, that means there won't be enough subsets to do RCTs in. There won't be enough people in all those subsets. And so many things get issued to NHS England for funding these days, and they say, well, they're not the cohorts. Therefore, can I have additional funding because they're not in the same cohorts? Because they've actually got a genomic variance. We've never looked at it like this, but virtually everybody has got a variance of some sort and therefore needs to be looked at differently. And this is where sort of physiologically based pharmacokinetics comes in. This is looking at absorption and distribution and really working out how a drug might be used. And so loads of modelling to be done then. And then there's also PK and PD dose modelling, looking very much at the effect of the drug. So instead of pouring millions and millions into RCTs, which actually don't give us the answer, we do need to start looking at dose modelling, PK, PD, PK, and then looking at real-world data to evaluate the outcomes of patients actually on them. Just before I finish, I will touch on the last bit of this. This is all brilliant, thinking, right, we are going to be able to define our populations by so many criteria, and therefore everyone might need a different dose. And that's really, really important. And you can almost see at some point in time, there'll be a little blood sampler just next to your bed, and you'll be able to do your drug levels, and loads of data will be in there, and you'll get your level out the next morning. And oh, hold on, something's changed. You actually need to increase your dose. And so there's a way that we need to be able to monitor and review what we're doing. Almost the things we used to think about with narrow therapeutic drugs. Well, in fact, do we need to be thinking of all drugs in that sort of realm? So how might we look at this? We could obviously put everybody on liquids, and so that everyone can adjust the dose. That would be an absolute nightmare. But there are other ways forward, and I think this is where life becomes very exciting and, again, interesting things to look at. Next slide, please. So how do we make sort of individualized dosing a reality? So next to that mini mass spec next to your bed, which says you actually want a small increase in your dose, do you have a little 3D printer which can print out an extra tiny bit of a drug? So instead of saying everyone needs 5 milligrams, you actually might need 4.75 milligrams, and it can print it out. The technology is already there, and these tablets can be made with coatings on, which make them sustained release. There's even a thing called 4D printing now, which is looking at sort of a variable time release. It actually remolds itself once it's at different parts of the gastrointestinal system, and so works very, very differently to conventional drugs, and releases far more effectively in the right areas of the gut. Are we worried about the whether we actually know where each drug's come from? Well, you can actually put a QR code on every single tablet that you print. And if you really want to do this for pediatrics, as it shows there, you can have your tablet in the shape of a teddy bear and have them chewable. You can do dinosaurs. You can do anything you like in any color. It all sounds very simple. It's obviously not, and we're quite a long way from this being the norm. And hopefully this will never be needed for every drug, going back to that wide therapeutic range, really wide, because standardization, I think, will always be, hopefully, the backdrop of medicine practice. Otherwise, it's going to be hard. But as we do move forward, there are a lot of drugs that do need specific doses. Drugs that we thought didn't work because the dose wasn't quite right, maybe they can work if we get the dose right. Someone's getting too many side effects. Maybe we can adjust it a bit, still get the good effect, but get rid of the side effects. And what we need for that is to get the real data and have mechanisms for producing the right sort of medicines at the end of this. Next slide, please. I hope this has been of some interest. I say it's not a pediatric discussion at all, but it is talking about the fact that personalized medicines are now a reality. And somehow we need to work out how we're going to deal with that difference in data. What is real evidence? And it's what we've always thought of being good evidence, actually not the pinnacle of evidence. And how are we going to deliver good personalized medicines to our patients? Thank you very much.
Thank you very much, Steve. That was really fascinating. I think food for thought on understanding or recognizing the individuality in all of us and how drugs need to be personalized to suit that so that we can optimize healthcare. There's some comments in the chat about the concept being really good in theory. So I thought I might relate that to a question, which is, we've understood the experience of a child. You've elicited that in your presentation, and we know that meds are relatively well understood in the adult population. What would you say are the biggest challenges in implementing a gene-based personalized medicines approach, and where are we in the journey of realizing its value? Because in theory, it's a great theory, but implementation is going to be a rather big challenge, isn't it?
It is. We're at the start of a journey, and I mean, just to comment that we've probably got a lot of it right in adults. I suppose the real question is, have we? The expectation, obviously, you know, going back to a lot of people don't take their medicines anyway. Therefore, what does that really mean? But by understanding real-world data, we will know far more about what is really happening. And it's a bit like somebody who takes an anti-epileptic for a long, long time and they seem stable, and then all of a sudden they do have a fit. Well, was the dose actually right? And it's easy to say we've got things right because people don't have problems for a long time. That doesn't mean we have. And I think we're getting to a stage where our understanding of drug levels, and whether that's kinetics, dynamics, genomics, is becoming more and more understood. And if we really looked at the population outcomes from everything, we would probably get a very different perspective. And this is what's being seen by the real world data. We'd get a very different perspective of how good we really are at dosing our patients. But we're at the start of a journey. It's a long way to go, and I don't think overnight things aren't going to change. But I do think it's a field that we need to understand more and more. And it's very easy to see that in sort of certainly 30, 40 years' time, and obviously we might not be in our lifetimes, probably won't be in mine, the way medicines are used is going to change.
Brilliant. Thank you very much for that, Steve. Really enjoyed your presentation, and we will channel any further questions to you. Thank you for having us. So without further ado, we will move on to the next presentation by Dharmisha. So we want to delve a little bit deeper in understanding the diversity in our sciences. So thank you, Dharmisha.
Hi, hope you can all hear me okay.
Yep.
So my presentation today is why we cannot ignore race when it comes to pharmacogenomics. So next slide, please. So pharmacogenomics is what I'm talking about here is how your DNA can be utilized to guide treatment in terms of precision therapy, in terms of drug gene matches and pharmacogenetics. So to understand whether a particular drug needs to be altered for you in terms of increasing the dosage, reducing the dosage, or that you need an alternative treatment. And so there are some pharmacogenetic tests which are available on the NHS, and we know that across a spectrum of diseases such as cancer and cystic fibrosis, there is some form of precision therapy happening. But what I'm going to focus on in particular is the diversity in pharmacogenomics, looking at the basic principles of how we should not ignore race when it comes to pharmacogenomics and what we can do better to make sure that we do not widen health inequalities or disparities when we're implementing new forms of looking at medicines and dosing patients. So next slide. So within this presentation, I'm going to be thinking about improving diversity, the considerations around it, and I'm going to use a case study called for DPYD gene to illustrate this example. This is a gene, a pharmacogenetic test that is available on the NHS and has been used in cancer for a little while now. And then I'm going to focus on some of the challenges of actually implementing this within the NHS setting. So I'm going to be talking about this from an NHS point of view, but this is going to be in terms of primary, secondary and tertiary care. So it's applicable to everything, and the Genomics Medicine Service Alliance is looking at all sectors in how we can implement this. Next slide. So let's start with how do we define and record? So when it comes to increasing and improving diversity in our science, what does it mean? So differences between people can be described in terms of race, ethnicity or ancestry, and all have been used interchangeably in the scientific literature to group people together. And it also depends on what papers you're reading from and from which country. So for example, American papers can define people as white, black or Asian. And then UK papers, it can be European or Asian, it can be African. So you get a bit of a wide range, and sometimes Asian can mean Indian, or can also mean Chinese. So it's really kind of constructed in different ways, and you really have to look at the data by what they mean about that and where the paper is being published. Race really is a social construct. It does not represent biology, and it does not capture the rich diversity within populations. So, and therefore you can think about, well, ethnicity can be used, but it broadly groups people together based on religion, food and language, so it may be more useful, but subtle differences within populations can still be missed. And this is why sometimes we need to think about ancestry a little bit more compared to the other ways of defining people. So next slide. So when we think about increasing and improving diversity, if we just take one element, Asian, for example, you can split them into different groups of people, from East Asian to North Asian, and even within those groups, they can be categorized in more individual forms as well. So what does that actually mean, and why is that important? So if we take another example, so if we go to the next slide, if we look at the word black, for example, as you can see, there's a huge range of diversity here. And if we take South Africans, for example, they can even be categorized in a different way. And this is all dependent on ancestry. And all of this has an implication when we're looking at pharmacogenetics in terms of guiding drug dosing. And the reason for this is, if we go to the next slide, that there is a huge range of heterogeneity within the continent of Africa, and it's more heterogeneous compared to other countries such as India or Japan, or even the UK, for example. So therefore, there's a lot to think about. So if we go to the next slide, We really need to move away from thinking along the lines of racial and ethnic constructs within pharmacogenomics and really focus on genetically determined ancestry. And so this will help us to guide how we can better optimize prescribing in terms of drug safety and efficacy when it comes to looking at someone's DNA and producing a particular drug dosing or guideline for that particular group. Next slide. So why is this important? Well, there is more genetic diversity and within the frequently used definition of African populations compared to Asian populations. And if we take one well-known enzyme, and if we look at the gene test for this, which is CYP2D6, a pharmacogene, it metabolizes 25% of all drugs that are in clinical use. But this particular gene is very complicated, and it's really complicated to sequence as well. So this gene for CYP2D6 has more than 100 different variants, and this can be across the populations across the globe. But in Africa, it has the most heterogeneity. So if we're using the word black, for example, it does not capture the rich diversity or the variance when it comes to looking at drug dosing or guidelines. And so if this is not picked up in research, it can be missed. And sometimes it could be an important piece of information that may be missing. Next slide, please. So if we take a little bit more look at this, so if we look at CYP2D6, ultra rapid metabolizers, so these are people that will metabolize a drug rapidly compared to someone who doesn't have a particular variant. This data indicates that 28% of North Africans have this variant, and they'll be classed as ultra rapid compared to only 3% of African Americans. So highlighting that there's even significant differences within a particular population, which would not be able to decipher if you simply categorize someone broadly as black within a research paper. Next slide, please. So what are we doing? Well, in 2021, it was reported that 86% of genomic studies have been conducted in people of European ancestry. And this tends to be across all types of research, not just in genetic research. And really, this data has not improved from previous reports in 2016, where 81% of genomic data or research was conducted in European ancestry. So there is this persistent bias that we can see within research papers, which then will implement the clinical care that we deliver. Next slide, please. So if we think about in the UK, the 100,000 Genome Project is a UK scheme to sequence genomes from around about 85,000 NHS patients. And these were people who were affected by rare disease or cancer. It actually includes 85% of people of white background and only 0.8% of participants who really identified as black. And this was self-reported ancestry. So this lacks representations within trials, which means that we cannot really understand the full range of variants indicated in different health conditions. And therefore, it means that we won't be able to understand the need for dose reductions, for example, or help understand how we can implement a particular guideline if we were to put a particular test on the NHS. And therefore, if you are assuming white European genetics broadly applies to everyone, it can ultimately lead to patients being mismanaged. And so the 100,000 Genome Project now is looking at diversifying their data set using a diverse data initiative. And there's also a new initiative called Our Future Health to actually ensure that the population that we are testing or putting towards research is actually representative of our diverse UK population. And if you saw the last consensus that came out this year, we really are a very diverse country, and we really need to make sure that everyone is represented within the research. Next slide, please. There's also a change that is happening in our society as well. There's a shift in population demographics. So increasingly, the number of younger generations between 18 to 24-year-olds, so this is from UK Gov data, actually identify themselves as belonging to two different ancestral groups. And this is different compared to those who are over the age of 60 who self-identify within one group. So there is an increased population admixture, and this can lead to greater diversity and possibly a greater genetic variation in pharmacogenetic testing, which is not commonly seen in certain populations, which is homogeneous. And admixed populations are also underrepresented in human genetic studies. So when we compare to homogeneous populations, Admixed populations actually provide a really unique opportunity to capture genetic architecture, which contributes to really multiple sources of population, and in turn can actually improve statistical power. So it's really important that people from admixed populations are approached to be part of studies as well and research. Next slide, please. So how can we improve? First element is building trust. There's a lot of kind of data, research that happened in the past, which is very checkered. So there's very checkered history in terms of Black African ancestry, where you've got the Henrietta Lacks and the Tuckasee syphilis experiment, or even now, if you had a look at the sickle cell report that came out recently, there is a lot of element of building trust within healthcare sectors and in research as well. In non-European individuals, there are reports that actually individuals are not being asked to participate in studies. There is a thought actually that they're discouraged. People feel discouraged of putting people from different ancestries into clinical trials because it leads to additional time and effort into reaching communities because of possible language or cultural barriers. And I think that's really important to highlight here. Community engagement has to have diversity in mind. So it may mean working with community leaders or thinking about language. So you might be able to change something technically, but you also have to do cultural translation as well. And also thinking about collaborative approaches and working with others. So for example, when it comes to genetic data in the continent of Africa, there is ways that we can work with the African Pharmacogenomics Research Consortium, so which aims to consolidate pharmacogenetic research in sub-Saharan Africa to accelerate translational into clinical application. But it's really important to note that if we take the data from people of African ancestry or minority populations, that we just don't take that data, and therefore if there's treatment that's come along, that they don't have the chance to access that treatment, so that it looks like exploitation in a way. So we really need to make sure that from the point of we're doing research to the point where the drug is available or a particular test is available for a drug, that we're really making sure that there's no health inequities across that spectrum. Next slide, please. So the ways that we are improving at the moment is not only do we have the Diverse Data Initiative with NHS England and Genomics England, but we've got the NHS Future Health, but there's actually more work being done or working with collaborative groups on a global scale with African genomic research consortiums, and these are just a list of some of them. And if you go to the next slide, please. This particular research group is called the H3 African Project, and their aim is to sequence 3 million genomes across Africa. So there's a way of doing collaborative working to make sure that the evidence and the information that we generate is really reflective of more of a global population and not just of white European ancestry. Next slide, please. So I'm going to be using DPYD as a case study to really look at equity, diversity and inclusion, and to really highlight some of the elements that I've talked about and bring them together as a case study so that you can see it in practice. Next slide, please. So on the National Test Directory, so we have two test directories. We have one for cancer, one for rare diseases, and DPYD is tucked into the cancer test directory. So DPD, it stands for dihydropyrimidine dehydrogenase, and this is an enzyme. And this enzyme is coded for by DPYD, which is a gene. So DPD is involved in the metabolism pathway for 5-fluorouracil, capecitabine, and tegopher. So these are anti-cancer drugs prescribed predominantly for gastrointestinal, breast, and head and neck cancers. So DPD deficiency is often caused by inherited variants of the gene, which are called germline mutations. So these are variants or genes that you inherit from your mum or your dad. And if you have a particular variant, patients who have a deficiency because they're not producing enough enzyme because of their genes, with this medicine, it increases the risk of serious toxicities such as stomatitis, diarrhea, mucosal inflammation, neutropenia, so that's low white cell counts, and neurotoxicity. So if we go to the next slide, please. So evidence for this particular test on the NHS is based predominantly to 4 genetic variants, which are listed in this middle part here where it says gene, and the 4 variants tested here. And the population study is from white European ancestry, so European population. So data from other populations are actually lacking. Next slide, please. So within North Thames, and this was done across the 7 GMSA sectors across England, we did a lot of work. Next slide, please. But the biggest element that we did was education and training. So really to raise awareness, not of the utility of pharmacogenetics and the advantages of it, but also the limitations of it. And really to provide educational sessions. And what we've done across North Thames is to highlight the limitations of this particular pharmacogenetic test. So it highlights areas that need more work. Next slide, please. And the case that I actually provide is this. So because the research is linked to 4 DPYD variants in white European populations, there are some other variants, such as in African ancestry, that could be placed on the test directory, but more evidence is required, and that is on the 3rd bullet point. There's a genetic variant there that I've listed called Y186C, and it's relatively common within African ancestry of 3 to 5% carrier frequency. It's currently not tested because more evidence is needed, but there are case studies that are emerging. And this is one here of an Indian patient. So she was treated with capecitabine for a GI cancer, and she was tested with the 4 routine DPYD variants that we test on the NHS. It was identified that no dose reductions were required, so she was treated with full dose capecitabine, and then subsequently she was admitted for severe mucositis of the ileum. So the doctors were wondering what was happening here, so they did further sequencing and they identified a rare DPYD variant, which was not documented before, and it could be not documented because we probably have never tested and we don't have the evidence for it, and it's probably because there's not enough information in the Indian population. So this was interesting because she obviously needed a dose reduction. So now there's work being done in India, so there's an Indian research group looking at DPYD variants in the Indian population. So there is a way of working with researchers in other countries to actually help guide us in this country in terms of the variants that we can be picked up. Next slide, please. This is a really busy slide, but I don't want to go into too much detail, so I'll talk you through it. So as I said, that research is linked to the 4 DPYD variants in white European populations, and these are the 4 variants below. But on the chart, on the table below that, the second table, we've got a list of other variants in different populations. And it gives you the frequency of each one. And if you go to the next slide, please. This is the variant that I picked up from the 3rd bullet point from the previous slide. This one is common but not frequently tested. So what we can do is, this is a data set that can actually be updated, and as more research comes across, we can actually look at the frequencies again and then decide whether on the NHS this is actually cost effective to do. So this is really ongoing work that is happening. Next slide, please. So what are we doing now? So we had a transformational project for DPYD gene testing on the NHS across England, and this we had a really big project in terms of national baseline surveys. We did a snapshot audit in terms of race and ancestry, and we also looked at ethnicity, but we really want to move forward because what we found out that ancestry or ethnicity isn't documented in patients' notes very well. So we want to repeat it in London, this particular audit, looking at a retrospective audit at a national level as well, to obtain more detail on how we can use real-world data on ethnicity and ancestry, and whether we can get more data in terms of toxicity. So there's more work being done. It's actually a deliverable in NHS England with the Genomic Medicine Service Alliance to look at ethnicity. And it's going to be a really collaborative effort, and we are working with devolved nations as well. So the work doesn't stop here, even though the test has been implemented on the NHS, because we are pretty much aware of the limitations. Next slide, please. So it raises to a next question, why are we not reaching the populations within research projects, what's happening? And it raises the question of how do we reach out to underserved populations? So really we're thinking about the inverse equity hypothesis and underserved populations. So what are the barriers to healthcare, and among them, who are the last to benefit from innovative health technologies? So pharmacogenetics is an innovative technology, and how should it be used so we don't increase disparities further? And this is really looking at the demographics, the socioeconomic status, and geographic regions as well, such as the deprivation scores by region. So that can be different from rural areas to cities. And underserved populations doesn't just include minority people. It could be people who are asylum seekers. It could be people who are unemployed, for example, or even people who are in the prison sector, for example. So it's really thinking really broadly about underserved populations. And I think when we're talking about EDI, we should really think about it in a really broad way. Next slide, please. Within North Thames, I'm actually looking at deprivation scores to help guide. So, for example, in Northwest London, it's a very diverse area of 2.4 million people from 200 ethnicities. There's differences in life expectancies, and we know that the most deprived area is Hillingdon. And this was taken from ICS data. But what's important here is that 28.6% of people do not have English as their first language, which is compared to 8% nationally. So if I was to do a patient information leaflet or from the Genomic Medicine Service Alliance, I really need to take this on board, and sometimes looking at deprivation scores and looking at ICS data can really help guide how we can deliver services. And particular work such as the DPYD project have been done with patient and public involvement as well. And we have an EDI strategy as well to make sure that we're thinking about all components when we're implementing a new pharmacogenetic test on the NHS. Next slide, please. So what are the considerations and challenges of pharmacogenomic implementation? So I've mentioned one, and I think there are a few more to touch upon just to include everything. So next slide, please. When we think about pharmacogenetic testing, we really need to think about the timing of pharmacogenetic testing. So we have a project in the North Thames about pharmacogenetic testing in psychiatry, looking at antipsychotic medication. And there's different ways you can test patients, and you have to think about EDI through each of these projects and how you reach people. It could be a reactive approach. So this is the most common element used in psychiatry and also in cancer, mostly in psychiatry. We can do it from a point of care approach, but more commonly, particularly in cancer, we use a preemptive approach. So this is where you have time and you know what treatment you're going to give to a patient. But each of these elements where you put a pharmacogenetic test will need a different way of engaging with the community. Next slide, please. So these are all the genes where there is evidence of a particular pharmacogenetic test. And if we just look at two of them, so if we go to the next slide, please. For CYP2C19, and the next slide, please. CYP2D6. These are drugs that are used. These, I should say, genes, CYP2C19 and CYP2D6, are predominantly used in pharmacogenetics in terms of antipsychotic medication. And there's a lot to think about in terms of guidelines because not all of these have robust evidence. So, and not all of these have good ethnicity data to back it up. And most of the data and guidelines are based, again, on European ancestry, not just in cancer. Next slide, please. So there's lots of things to think about when it comes to hurdles. Not only are we thinking about ethical and legal issues and diversity, there's also about cost effectiveness and standardization, and also test cost and reimbursement, and also how you integrate some of this information into electronic health records. So next slide, please. And these are the main elements that I've thought about in psychiatry, but also across pharmacogenetic testing. And it's most of it, particularly about improving diversity in pharmacogenetics. It's about knowing the limitations of testing and also the knowledge and the education gaps we need to fill so that we can utilize these tests safely within the healthcare sector. Next slide, please. So this is my final slide. How are we ready? The key considerations when we think about pharmacogenetics is not just about like pharmacokinetics, pharmacodynamics, which you know, but the genetic data, the environmental impact. If we were really to get personalized medicine, we need to think about the cost effectiveness, the clinical guidelines, the reimbursement issues, education and training, the delivery of this. And when we think about our diverse cohort that we have within the country, we need to bring patients with us, and we need to have strong data based on that includes everyone. And also when it thinks about quality control, if we don't have that data there for a particular ethnic minority, we may not be able to get it onto the NHS if we don't have the data to support it. So next slide, please. So pharmacogenomics, it's not going to replace our clinical judgment. It's an additional tool that we're going to use in our medicines optimization toolbox. And we still need to consider the key things that we do all the time, such as read on liver function, drug-drug interactions, comorbidities. But when we implement in a test, we really need to understand the limitations, which I've mentioned a few. But if we're to bring everyone with us, we need to make sure that everyone is represented in the research so that when they are available on NHS, that the service that we provide is truly representative of the population that we serve. And that's my last slide.
Thank you, Dharmisha. That was really informative. It gave us a really good understanding of the different types of racial groups, the various subtypes and considerations for the permutations and combinations of what we need to consider to personalize medicines, really. In the interest of time, we've got some questions, but I may not be able to post them to you right now. So I'll wrap up the masterclass, and we'll be able to reach out to our listeners with the questions. So thank you very much. So I just wanted to wrap up by saying today has been really insightful. We've been able to understand the practice of evolving from our current model of healthcare towards more practice-based evidence medicine, as shared by Steve. We've also been able to understand the uniqueness of our diversity in science, as shared by Dharmisha. I just wanted to reach out to all our listeners to say we'd like to thank you for your support in attending these webinars, and we would love to hear from you about any future topics of interest. I've put a link in the chat for you to be able to engage with us from the perspective of digital transformation, or even if it's more targeted topics and methods to optimize your EPMA systems. We have a community of practice tab for you to discuss any topics, and we will actively monitor it. Alternatively, you can reach out to me with my email or my Twitter feed there. So just to summarize as well, our next masterclass will be on the 8th of March, 2023, between 1 and 2 p.m. And with that, I will wrap up the session, and thank you all for coming again. I wish you all a great Wednesday. Thank you.